Hi dear students, welcome back to one more session of genetics. Previously we had come across the Mendelian laws of inheritance, the law of dominance, the law of segregation, as well as the law of independent assortment of genes. In the law of dominance, we found out that one of the particular trait or one phenotypic trait of a particular uh, character has been found to be dominant over the other. Like you have tall as well as dwarf, in which tall has been dominant over dwarf, and that was a law of dominance. And the one which was less expressed or which was being hidden was referred to as recessive nature. And we also came across a law of segregation in which the different alleles get segregated during the gamete formation. And we also came across the law of independent of assortment of genes, in which when we consider a dihybrid crosses, the different alleles of different traits happen to independently assort during the gamete formation, and they could independently assort and form different gametes independent of their occurrence. And that was the law of independent assortment of genes, as we noted in the Mendelian genetics. But we find that in spite of all these laws of Mendelian laws of inheritance, there do exist various variations for this Mendelian genetics. And sometimes at least you can find that some genes are being found to be more predominantly expressed, and some alleles are being found to be closely associated with each other, that they do not segregate also, or they rarely segregate also, as in the case of linkages. So as I mentioned earlier, we're going to explore linkage and crossing over. Before starting into the core part of the study, I would like to take you to a small game in which we are crossing sweet pea varieties. We are going to do a dihybrid cross of sweet pea, because it contains two types of flowers. You have purple color flowers, as well as at the same time they have red color flowers. And if you go to look into the pollen grains of these sweet pea, they can occur in two different forms, one as a long form as well as in the round form. Hope so you know what is a pollen. I have shown you a dissected flower here, and you can see on top of the fourth one, that is the stigma, and the pollen is something which is coming from the anther. And so the deposition of this pollen grains is onto the stigma plays an important role in the fertilization and the development of the particular plant. And when you go to look into sweet pea, it has mainly two different types of pollen grains. Either it would be long, or it can be found to be as round forms. So we are taking two main characteristics here. One is the flower color of sweet pea, as well as the pollen grain, which could be long in the type of wild variety, or it could be round, as we have shown here. And when you cross these two type of strains, you can see that, or when you can conduct a dihybrid cross here, you can see that you would get a hybrid variety of capital letter P, small letter p, capital letter L, small letter l. And unlike you can see that the parents, though they have been found to be violet and long, you can see that the hybrid is also been found to contain purple as well as long type of varieties are being obtained. Now, from this cross, we can identify that the purple flower and the long pollen grain are being found to be dominant characters, and they are being expressed in the hybridized or in the F1 generation. So here, the purple flower is dominant over the red flower, as well as the long pollen grain is being found to be dominant over the round pollen grain, and that is the reason why the F1 generation is being formed with the purple flower as well as a long pollen grain. If you go to do the next step, when you do a test cross between this F1 hybrid with its recessive parent, we are mainly doing a test cross. And you can see the possible gametes which can be formed are being put in a Punnett square over here. And this experiment was also being done by Bateson and Punnett themselves. And we are just repeating what they did. And you can see that these are the different varieties of gametes which have been formed here. Now in the F1, from the F1 hybrid, you can find capital letter P, capital letter L, capital letter P, small letter L, as well as small letter P, capital letter L, and small letter P and small letter L type of gametes are being formed. As well as from the recessive parent, you can see small letter P, small letter L are the gametes which are being formed. As per the Mendelian genetics, we can see that these gametes which are being formed during the test cross, the usually expected result is that we will get purple flower long pollen with a long pollen, purple flower round pollen, as well as red flower long pollen and red flower round pollen in the ratio of 1 is to 1 is to 1 is to 1. That is a common ratio that we expect as per the Mendelian laws. But Bateson and Punnett, they noticed that this was not the exact thing that they noticed. There was a slight variation among the results, unlike what they expected. The actual results showed that there were purple flowers with a long pollen in in a ratio of 7 is to 1 is to 1 is to 7. That is, the purple flower with the long pollen, the purple flower with the round pollen, the red flower with the long pollen, as well as the red flower with the round pollen were being found in the ratio of 7 is to 1 is to 1 is to 7, unlike the expected 1 is to 1 is to 1 ratio as per the Mendelian law. So we can see that during this study, Bateson and Punnett, they noticed that the parental characters of purple flower with a long pollen as well as a red flower with a round pollen were being found to be predominantly in number than the hybridized ones which have been formed. So they were thinking, what was the reason or what was the exact reason behind this? We'll explore that. Now, why did such an occurrence happen and how can it be explained? Bateson and Punnett, then they put forward a hypothesis of coupling and repulsion to explain this. And they told that the tendency of the dominant and recessive alleles to inherit together was explained as gametic coupling, and the tendency of both the dominant and recessive alleles to repel each other, so that the non-parental or the recombinant gametes were formed as was termed as repulsion. And they do not know they could not explain the exact reason behind this phenomenon, why the recombinant gametes or the non-parental gametes were being formed less frequently. But they told that probably this reason why the hybrids or the non-parental gametes were being formed in a lesser concentration than the parental gametes could be as a result of gametic coupling as well as gametic repulsion. That's what they made a hypothesis. But the exact reason for this or the scientific basis of this was linkage, and that was of course put forward later on by Morgan. And so the further experiments which was being carried out by Morgan led to the discovery of linkage.